Thank you so much for inviting me. It's a real treat to get to come back here to Stony Brook. When I was young, my aunt and uncle had a house at Port Jefferson, so it was really, nostalgic coming back here to see it again. Today, I'm going to be talking about biology of emerging contaminants, and I hope we can ponder whether they really do eventually emerge or not. And I think the answer that we'll come to, not to steal the thunder, but is some do, some don't. And I also want to give a special thanks to Jinwei Mao, professor, who was one of my PhD students, and I'm so proud of her. When she was in my lab, we did all kinds of interesting work with chlorinated solvents, which at the time were still things that were semi-emerging, but they're pretty established at this point. So let's move on. Today then, what I'd like to do is take us on a whirlwind tour of a number of different emerging contaminants. And I'm gonna take us through 5 different case studies: MTBE, NDMA, 1,4-dioxane, PBDs, and PFASs. And yes, I did not spell out any of those acronyms on this slide, but I'll spell them out when I get to them so you don't forget. And what's interesting about this interesting student interlude is every single one of those emerging contaminants came about because a student came into my office and said, hey, I just read about this chemical and nobody knows whether it's biodegradable or not. What do you think? And we said, all right, well, let's give it a try. And so that's how all of these began. So the list of emerging water contaminants grows continuously. And as you can see there from the total pubs and the total citations, exponentially. And the hypothesis we have for why this happens is, 1, we're continuing to discover new contaminants because our analytical techniques are just getting better and better and better. We're able to measure things at much lower concentrations, and so we're getting better at finding the contaminants, and that's part of the reason. We also have increased water demands because we're not increasing the amount of fresh water on the planet, but we are increasing the number of people on the planet. So those increased water demands results in the need for us to use more impacted sources, and therefore we need to clean them up and we need to know what's in them so that we know what we're being exposed to. And then direct and indirect reuse is becoming much more common. Again, we need to know what we're effectively removing from our water and what's being left. So back to our outline, let's jump right into MTBE. Methyl, tert, butyl, ether. Nice little organic compound there. You can see the ether with yeah, that's not gonna work for me. With that, oh, there it is. With the oxygen there in the middle. It is an oxygen that was added to gasoline in the 80s and the 90s, and it was added to curb smog, because smog was a very, very large problem for us at that time. Why MTBE? Well, it was cheaply produced from waste products of petroleum refinery industry, and it became the 2nd most produced chemical in the US in the 1990s. Think about that, the 2nd most produced chemical. That's pretty impressive. It's hydrophilic, it loves water, it moves right along with the groundwater. It absorbs poorly, and it was at the time thought to be recalcitrant. The reason people started to worry about it is, as you can see in those depictions there, we have underground storage tanks, and there are 2 types of underground storage tanks, those that leak and those that will leak. Every gas station has like 3 types of underground storage tanks, right, for your regular premium and whatever they call the elite one. And those tanks eventually spring leaks. And so the gasoline gets into our water. Gasoline is made up of mostly biodegradable organics. MTBE, which is hydrophilic, would move then from the gasoline plume and move by itself and become its own contaminant. The other thing is that it has a taste and odor guideline for drinking water of something on the order of 20 to 40 ppb. That means a dropper of MTBE in an Olympic-sized swimming pool, and you'll be able to taste it and smell it. So that's part of the reason it became a huge problem for us. So how was it chosen as an oxygenate? Gasoline producers thought about economic issues. The other oxygenate that they were considering was ethanol. MTBE they could make in-house by themselves. Ethanol they would have had to buy from farmers. So that was one thing. MTBE blends easier than ethanol, has a higher octane rating, and was already approved for regulatory use for small amounts. So they just increased the amount of it in our gasoline. It had 15 minutes of fame. In the early 90s, California had introduced reformulated gasoline 2, went into effect, and it contained MTBE as an oxygenate. And the San Francisco Chronicle writes, all of California should welcome the new gasoline like a fresh of breath air on a smoggy day. And then the infamy. In the 90s, USGS began to start measuring MTBE in our urban wells and shallow aquifers. It was found in many aquifers in California as well as Maine. And I don't know if you guys are old enough to know who Steve Croft is, but of 60 Minutes fame. If you don't know about this yet, you will. It's a gasoline additive that is contaminating water from Maine to California, and it has been called the biggest environmental crisis of the next decade. Hyperbole much? I mean, honestly, compared to global climate change? All right, never mind. The good news is in the late 90s, we did a lot of work on this. Not only our lab, but a bunch of other folks worked on it as well. And we found MTBE to be biodegradable by a variety of different microorganisms, both metabolically and co-metabolically. We went on to show that we worked out the transformation pathway that you can see over here. It's pretty simple because it's a simple molecule. And we showed how it could be inhibited by the presence of some components of gasoline, but not others, and that how MTBE's present inhibited some of the degradation of gasoline products, but that both of them could eventually be degraded by the same microbial communities. So conclusions, and Rula Deeb was the student who ran into my lab and introduced this topic to me there, and that's her photo. MTBE is degradable, both, I already said that, metabolically and co-metabolically, and we talked about the inhibitory effects. Bioremediation is possible, but it is challenging because getting the right microorganisms to grow can be a challenge, but it has occurred. And then elimination of MTBE in gasoline has significantly reduced its presence in our groundwater. Why? Because it moves so easily with the water, so you can flush it out pretty fast, and it doesn't have a long-term source. Okay, let's move on. NDMA. N-nitrosodimethylamine. There it is, a little molecule with a couple of nitrogens in it. It is a potent human and animal carcinogen. It's made from rocket fuel degradation as a byproduct. It's also used in plasticizers and as a solvent. It also, this was very interesting, it can be made by chlorination of water and wastewater and in membranes. We found out it can be made in membranes because in our ultra-pure water in our lab, we were finding trace quantities of NDMA, and we found that it was actually being generated within the membranes, which was very strange. At any rate, regulatory concerns are at the parts per trillion levels because of its carcinogenic properties. It's extremely hydrophilic, again, moves right with the groundwater, low volatility and poor absorption. Routes of exposure are occupational from rocket fuel production. Ingestion, you find this material in hot dogs and cured meats, cigarettes, of course, beer, unfortunately, and then cutting fluids, pesticides, and plasticizers. This is an example of an NDMA plume at an aerospace facility in the LA basin. Rocket fuel was processed there from 1943 to 1968. The concentrations were quite high, and you can see it's miles long, 7 miles as a matter of fact. So we looked at it and we had a hypothesis. We said, it seems to us that the studies that have been done to show that it's a carcinogen were done with rats and other mammals. And they showed that monooxygenases within the rats would metabolize the NDMA and turn it into even more toxic forms. And so maybe we can use the monooxygenases in bacteria to biodegrade it. And so we tested a whole bunch of monooxygenase generating bacteria to test for that. And what a monooxygenase does, they're very cool enzymes. What they do is they take molecular oxygen, break it apart, take one of those oxygens and stick it on an organic, and then the other goes to water. And so you end up then with usually the equivalent to alcohol from the original compound. So we tested a bunch of bacterial isolates that were known to have monooxygenases, and this was the work of Josh Sharp primarily, but with others who I'll show in a minute. And voila, we found that there were a variety of microorganisms that could be, monooxygenases were induced by propane or by methane or by toluene who are able to biodegrade NDMA. Those are shown in green up there. The ones that are shown in pink, I highlighted them pink because they also have induced monooxygenases, but those monooxygenases do not degrade NDMA. So the lesson there is don't just assume a monooxygenase is going to do it. It's got to be a particular type of monooxygenase. And then the ones in white were showing the negative control. As part of that work then, we used a very fancy machine to ferret out the degradation pathway for the NDMA and show that it is indeed mineralized. And then we used gene chips to evaluate the global gene expression, because what we were really interested in was identifying in these organisms which particular genes were involved in the degradation. So the conclusions from this work, and there's Josh shown graduating at the top. He's at Colorado School of Mines these days. And there is Chris Alvarez at the bottom, who is a professor at Drexel. And they carried out the majority of this work. They identified a variety of monooxygenase expressing bacteria that could degrade it, and specifically one that has that degrades it very quickly, which is this Rhodococcus RHA1. They then identified the monooxygenase in RHA1 and then cloned it out of that microorganism, put it into E. coli, and demonstrated for sure that that was the one that was initiating the degradation pathway. Okay, let's move on. 1,4-dioxane. I know some of you here are working on 1,4-dioxane. I look forward to hearing about those later in the afternoon. 1,4-dioxane is a diether shown there on the left. Your left, my left. Okay, shown there. It is not 2378 TCDD, which is dioxin, so don't get confused between those two chemicals. It's used as a stabilizer for chlorinated compounds, most specifically for TCA, but also for TCE, PCE. It's solvent for paper and textile production, and in the manufacture of organic chemicals. It's also a component, and currently still is, because it's not used for a solvent stabilizer anymore, but it currently still used as a component in cosmetics, shampoos, some fumigants, and some coolants. Probable human carcinogen, has all kinds of nasty effects. Regulatory level 3 to 300 ppb. So again, we went to our old friends, the monooxygenases, and said, okay, maybe they can do dioxane as well. And it turns out the two at the top in white, there we go, the pseudonocardia were able to biodegrade dioxane and, in fact, could mineralize it. That one at the top, CB1190, in fact, mineralizes it and grows on it, incorporates it into its cellular, into its cellular products, and I'll show you some data about that in a second. The second one is is also able to biodegrade it. So there are a couple of pseudonocardias that can do it. However, there are some pseudonocardias shown in pink there that cannot. So again, don't assume if it's a pseudonocardia that it can biodegrade 1,4-dioxane. Also, our old friend methylocyanus trichosporium, which is a methanotroph, can biodegrade it if it is induced with its soluble monooxygenase, but not its particulate monooxygenase. It happens to have two. So most but not all monooxygenases degrade it. All that do are inhibit are inhibited by acetylene. So acetylene can be a nice tool to use to see if it's a monooxygenase. And then E. coli recombinants with the toluene monooxygenase were able to degrade dioxane like the wild type, demonstrating that indeed we had identified the correct enzyme. That's what it looks like at the bottom. It's very hard to grow. It's got these long spindly things. It's called pseudonocardia because it's kind of like a nocardia, but it's not a nocardia. Nocardia is what you find in, it screws up your activated sludge units, makes them so your sludge doesn't settle well. These are similar to that, but they're not the same species. Shown on my side is the pathway that we worked out for the mineralization of 1,4-dioxane. And then shown at the top on the other side is C14 labeled degradation products from 1,4-dioxane to demonstrate that for the first 5 columns are 5 different species that were able to mineralize all the way to CO2, the dioxane, whereas the last 3 could biodegrade it. They could take it a couple of steps, but they couldn't mineralize it. We also were able to identify the monooxygenase in the CB1190 strain. We sequenced the entire genome and identified it. And we found out there are 8 monooxygenases there, and there's only one that does it, and that one that does it is on a plasmid. And that plasmid is a little piece of the chromosome that that microorganism can lose if it's not exposed to 1,4-dioxane long enough or continuously. And so we thought that that was an interesting element of the story. Shaylee Mahendra and again, Christopher Salas are the 2 people who worked on that. Shaylee was the one who ran into my office and said, hey, have you heard of 1,4-dioxane? So they identified the bacteria that degraded, sequenced the strain, confirmed the activity by expression in another organism. And then they also went on, this was Chris's work, to develop a model of the CB1190 central metabolism. He used labeled carbon compounds and fed it to the organism, and then were able to track exactly how that carbon was metabolized by that organism. Okay, how am I doing on time? Okay. PBDEs, polybrominated diphenyl ethers. These things are brominated compounds. They look like PCBs, but they're slightly different. First of all, they're used as flame retardants. They're in many, many common household things, like perhaps in the cushions and fabric that you're sitting on. Lots of rugs are treated with these things. And there are 209 different congeners. 209, because you can have all 10 of the carbons brominated, or you can have 9 of them brominated with 1 of them with a hydrogen, and then you can go all the way down to having only 1 brominated. And then if you do every combination of all those particular things, you get to 209. And so whenever you run into PBDs, you're dealing with 209 compounds at the same time. Those of you who are analytic chemists, how fun is that to separate out 209 different congeners? All right, they're extremely hydrophobic, so they stick to everything, making them really hard to work with. They have a low vapor pressure, and they're also in things like TV, computers, et cetera. You see the list there. I won't read them. They also, oh, by the way, since they're so hydrophobic, they bioaccumulate. So they build up in your tissue as you're exposed to more and more of them. Okay, so seen on the left, seen on this side, is a GCGC. It's a 2-dimensional gas chromatograph separation of the 209 congeners of PBDEs. This is a very specialized machine. We had a collaborator that was kind enough to allow Kristen to go to his lab and do a bunch of testing for these. At any rate, using them, we were able to then feed them to anaerobic microorganisms. Here's where the work that Jinwei was doing, we fed off of that, said, okay, let's see. With PCBs, we know that anaerobically, they can be dechlorinators by dechlorinators. Aerobically, they don't do much. And so maybe PBDEs can be anaerobically debrominated by dechlorinators. And so we fed them to dechlorinators, and shown in the columns over here are the different congeners as they're going from the deca, from 10 fully brominated down to less and less brominated. And this is a summary, and I'm sorry I'm running through a lot of data, but I'm not actually showing you the data, just showing you the summaries, but this is a summary of 4 different microorganisms. They're all dechlorinators, and it shows that they all treat PBDEs differently. So starting with the deca at the top, there was only one that was shown in our studies, but other people have subsequently done work to find slightly different results. But deca, that's fully brominated, all the way down to mono, that's the 1 bromine. So there were one organism that could take deca to hepta, and then one that could take nona to di. I have the little skull and crossbones by the penta and tetra, because those are much more toxic than the ones on top. The ones on top are much more inert. So what you don't want to do, for sure, is you don't want to debrominate partially and get to the really toxic ones and then leave them there. You want to make sure you can get past that. And so they could take the nona or the octa all the way down to the di. So that's 2 bromines. And we also worked out the pathways for each of those different microorganisms and how they did it. And I could give you some trends there. But like PCBs, in the PCB literature, we found that first you dechlorinate a little bit, and then you swap it to aerobic conditions, and then it can break, the microorganisms can break the ring because they can get at it, because you've gotten those pesky chlorines off. So we figured we'd try the same thing with the PBDEs, and in fact, now I'm switching to aerobic conditions, and under the aerobic conditions, we tested 4 different aerobic microorganisms. In fact, you might recognize some of those names from the other studies, because they were the ones who were doing MTBE and 1,4-dioxane. As you can see, the biodegradation of mono can be carried out by all 4 of them. So aerobically, if you get down to one bromine, aerobically, a bunch of different organisms can biodegrade it. But several of them could take it all the way back from penta and tetra as well. And so the story there was you could do the 1-2 punch. So there's Christian Robrock and J.Z. He, who were the 2 people who pioneered that work. What we learned, PBDs can be debrominated by dechlorinating bacteria. The pathways are similar for all tested bacterial species. The meta and para positions are removed more easily, similar to what we observe in PCBs. And mono through hexa PBDs can be degraded aerobically, but, and this I didn't mention back there, so I'm glad I have it on this conclusion slide, the anaerobic process takes months. Weeks to months, but really it's months. The aerobic process, on the other hand, is very fast, goes within days. And so when you do that polishing step, it can be much faster. We can get complete breakdown from hydroxylated PBDEs. And then also as a side, which I'm not showing the data for here, we showed that photolysis can also be used as a destruction mechanism for PBDEs, and it's much faster than biodegradation. Okay, now PFAS, per and polyfluorinated alkyl substances. And I'm so excited to see that we're going to be hearing a lot about PFAS this afternoon, so I'm really looking forward to all your talks. I want to just set us up a little bit. We know extensive application of aqueous film-forming foam by firefighters, say that 5 times fast, has caused release of PFAS into the environment, and this has happened at a lot of DoD sites as well as firefighting practice sites, which is why CERTIP is funding a lot of research in this area, because our DoD has really had widespread contamination due to this. It's also, these are have widespread use in consumer products for waterproofing and nonstick surfaces, Teflon, Scotchgard, things like that, heat resistance, and et cetera. So our mission was to determine whether biotransformation can occur under redox conditions, and if so, to what? And can biotransformation be combined with chemical methods? So it's pulling us into the chemistry world. And then how to track the many PFAS compounds to determine where they're likely originated and what happens to them when they get into the environment. Okay, so this is meant to be a quiz slide. This was provided to me by a student who just graduated, Emily Cook. And the quiz is supposed to be, which of these pictures depicts something that PFAS is not found in? And the answer is, it's a trick question. They're found in all of these things. So it's hard to find things that PFAS is not present in, which is sad but true. All right, so what are these things? It's an alphabet soup, and I apologize for all the acronyms, but I've already mentioned that they're poly and perfluorinated compounds. If we look at the perfluorinated compounds, that means we are focusing on this tail here. And if there's a tail and there's as many fluorines on there as you can get, and then you have a head group, which can be a sulfonic acid or a carboxylic acid, or it could be a lot of other things. But if you have one of those two things and then a bunch of other stuff, then it's still a perfluoracid, but it's still a perfluoroalkyl substance, but it can be either one of the acids, which are the two, or is the one up that's showing there, and then the carboxylic acid that's similar to it. That's PFAS and PFOA. But we also have other things that are called precursors. So then there are a lot of other PFAS compounds that have crazy things after the tail, and then some that have crazy things before the tail. So this one here has, there you go, 2 carbons that are not fluorinated. So it's a polyfluorinated, it's not a per. And they're made by fluorotelomere production as opposed to electrochemical fluorination. So you can get a variety of these things. Here's the bottom line. The bottom line is there are thousands of these things that are known to exist. Literally over 10,000. And that makes it really, really difficult for us to be able to work with them because they come in these crazy mixtures. And so shown here, if you take the PFAS tree, then you break it into the perfluoroalkyls, the PFAS, the PFOA is shown in red, the PFCAs, those are carboxylic acids, the PFSAs, those are sulfonic acids, and then you also have the polyfluoroalkyls. There are thousands of them that are known. This is an old slide, so that 12,000 is already been blown by. Here's the crazy part. We have less than 100 standards. So quantifying these things is really, really difficult. And it is impossible for many of the compounds. So we have to think about ways that we can cluster them so that we can do some sort of quantification. I'll talk about that in a minute. Okay. So we started this work by testing one of the most common PFAS that's found in firefighting foams, which is called Lodine, or FTTAOS up there. It is one of a small component, it's a large component of the PFAS in the firefighting foams, but firefighting foams are made up mostly of water. They also have other surfactants, including organics that are biodegradable. So it makes it a very complicated system to want to measure. So we put together a bunch of microcosms. And we have, I work with David Sedlak, who's my colleague, who's an analytical chemist, thank goodness. He has fancy equipment. So we use LC-MS-MS to quantify them. We also quantify them using an assay that I'll talk about in a minute. The bottom line is under aerobic conditions, Under aerobic conditions, we are able then to biotransform this Lodine material in the presence of just your garden variety microorganisms from a contaminated soil. They aerobically are transformed. What are they transformed to, you should ask? And the answer is, under almost all these conditions, what I'm going to be talking about, things are transformed to the perfluorinated carboxylic acids and the perfluorinated sulfonic acids. So that's why we call them precursors, because everything that had all those crazy head groupings on them, those things get chopped off by the microorganisms, but they can't do anything with that fluorinated tail. And so you always send it to the PFAS and PFAS, and then the other ones that have lower carbon chain numbers, because they started with lower carbon chain numbers. And so under the conditions that have been studied so far, biotransformation is going to get you to endpoints that are products that we do not want to be exposed to, and that are, in fact, currently regulated. The precursors, because there's so many of them, and because manufacturers keep putting crazy head groups on them to get ahead of the regulatory, those are not currently regulated, but they're thinking of how to go about doing that. At any rate, so this shows both the tail with the 8 carbon tail, the 6 carbon tail, and the 4 carbon tail can aerobically be biodegraded. We propose a transformation pathway, and I won't belabor that. Then we tested under anaerobic conditions as well. Under anaerobic conditions, we looked at nitrate reducing, sulfate reducing, iron reducing, and then methanogenic. And as you can see, as you get more and more reduced, the reaction becomes slower and slower and slower. We also were able to show, well, I'll show that in a minute. Okay, so we recognize then that we're not getting very far by just looking at the biotransformation. So let's think about what we could do, maybe a 1-2 punch. Biotransform it to something simple like the carboxylic acid, and then figure out a way to transform the carboxylic acid in situ. That was important to us, that we wanted not to pull these things to the surface and treat them. We wanted to be able to treat them in the subsurface. So the method we chose to work with is persulfate. Persulfate, shown here at the top, can be heat activated, and when you do that, you produce sulfate radicals. And those sulfate radicals can attack the carboxylic acid, the percarboxylic acids, and pop off from the end, pop off each of those carbons with a fluorine. And so you go through a subsequent shortening of the chain until it mineralizes. And shown here is some data where we tested, because it had been shown that this happens at 85 degrees. We wanted to test to see if it could happen at lower temperatures that would be more conducive to in situ application. So we were testing it at 45 degrees C. And shown here is the concentration of the perfluorinated carboxylic acid all the way towards me. And then look what happens after the first application. After the first application, it pops up. You start low, and it gets very high. What happened? What happened was the first thing that the sulfate radicals attack are the precursors. And it attacks the precursors, chops off that head, and turns it into the carboxylic acid. So you're actually generating more of the carboxylic acids. Then, once that's done, then the sulfate radicals chop up those so that you get smaller and smaller chains. The different colors there are representing different chain lengths for the perfluorinated part. But we were able to show that, indeed, we could carry out these defluorination reactions with persulfate. And I wanted to point out, that the x-axis is in days. So it's not a very rapid process. We then went on to look at compare how the precursors fared biologically and chemically. And what we learned is the precursors in 3M, which tend to be sulfonated, as you can see the sulfonic head group there. Under biological processes, they biologically transform to the sulfonic acid, so that would be PFAS. And that's bad, we don't want that. But with this chemical process, with the, for those compounds, they were chemically transformed to the carboxylic acid, which we then could treat with the persulfate. So that turned out to be good news. And then the fluorotelomere compounds, which is represented here by the 6,2-FTS, under both biological and chemical processes, would go to the carboxylic acid. And so in that particular case, then that could be hit with the persulfate and still be an effective 1-2 punch to work on those. Okay, then the last thing I want to, well, one of the last things I want to say about this is I talked about the difficulty in quantifying. And so what we have to do is we have to cluster these things. So my colleague David Sedlak and his student Erica Hoots had developed this process where you take persulfate in order to quantify your PFAS compounds. You take persulfate, you add it to your sample, you heat it up to 85 degrees C and at a pH of 12, so very high pH. And when you do that, the sulfate radicals generate hydroxyl radicals, and the hydroxyl radicals then convert all of your precursors to the carboxylic acids. And so now you can measure the carboxylic acids, which we do, in fact, have standards for. So you don't know exactly what each of the precursors were, but you know what the entirety of the precursors of, for example, an 8-chain or a 6-chain or a 4-chain are. And so you can at least get a better idea of what you're dealing with. And it turns out to be a very effective method. So we wanted to use that method to see if we could develop mechanisms to determine when you come upon PFAS in the wild, how do you know where it came from? We have lots of different sources. Is there a way we can potentially fingerprint these things and get an idea of when you came from a wastewater treatment plant or when you came from a firefighting pit or whatever? So what we did was expanded the top assay to give us more detailed information about the PFAS compounds. And then identified data that could be useful to do source fingerprinting. So Ned Antel, who did this work, did most of this during the pandemic when he was not allowed back in the lab. And what he did was he combed through as much of the data that's been reported on chemical analysis of PFAS as well as top analysis. So a lot of people have been using top analysis. You can get it done at commercial labs now. So he put those things together to see if he could identify PFAS sources. Now, shown in the plot closest to me is all of his data plotted up for airports in blue, wastewater treatment plants in orange, and landfills in stars. And these are principal component analyses. And 95% confidence intervals are the ellipsoids that are around the data. And as you can see, without the top assay, the data is conflated. You can't really differentiate sources for those. But once you combine the top data with that data, you can see on the far plot that, in fact, they separate out pretty well. And now he has gone back and asked for samples from a bunch of different sites so that he can go ahead and further test this. He also turned this method into a website that anybody can go then and upload their data on and have this analysis spit out, okay, we think your data is coming from this kind of source. So it's kind of very useful. He just published that. So PFAS conclusions, we have more to come. We're still doing a lot of work with them. Major precursors in AFFF are biotransformable, that is, the precursors are, but not the fluorinated head group. They're transformable under aerobic, nitrate, iron, and sulfate reducing conditions. They are not mineralizable that we've seen. There have been a couple of studies that claim mineralization, and it's, you know, the jury is still out on whether that's going to be consistent. Transformation products are complex and elusive. Excuse me, PFCA common co-contaminants, TCE, dioxane, and BTEX. I didn't show this data, but when you have those those products together with the other compounds. They can be mineralized by both the persulfate reaction that I talked about, as well as by microorganisms that are transforming the precursors. So that's kind of some good news. But PFSAs cannot. That is the sulfonic acids. And then forensics of a triple F contaminated source can be facilitated by the application of the top assay. And all those beautiful smiling faces are the people who have worked on this. Okay, so last slide. Do emerging contaminants eventually emerge? Well, some do and some don't. MTBE, NDMA, and dioxane are not the problems that they were. I understand we still have dioxane problems, but they are not as major of a catastrophe as we thought they were. They're still important to when we encounter them, but we're really not encountering much MTBE or NDMA anymore because they were stopped being used and have pretty much been flushed out of the system. PBDEs and PFAS, on the other hand, we are going to be saddled with these problems for a very long time. And PFAS, since they're found everywhere, is going to be a major problem for us. And so congratulations to those of you who are working in the PFAS field. That is job security. Identification of contaminants and their potential toxicity is critical. So if things aren't toxic, do we need to worry about them? That's a big question. And, you know, figuring out how we're exposed to these compounds is a really interesting one. PFAS is a great example there. We look in all of our tissues and we find it everywhere, even in the tissues of newborn babies down in Brazil, right? How are they getting contaminated by these compounds? It's a big, really important question. I would never say never in biotransformation. It is amazing how many times we've said, oh, this isn't transformable, and it is. We said that about chlorinated solvents years ago, and it was. PBDEs the same way, never say never. Advanced molecular tools and others are allowing us to really shrink the timeline in how long it takes us to figure out what's going on in a system. So that's been terrific, and advanced analytical techniques as well. And finally, the vast metabolic diversity of microbes render them an important tool for addressing environmental contamination, but especially, and this is coming from somebody who's been almost exclusively an environmental microbiologist my whole time, but especially when combined with chemical and physical processes so that we can have treatment trains that get us to a complete solution for these compounds. And then I'll just leave up there the pictures of my lab and acknowledge all of their hard work because they do all the hard work. So thank you very much for your attention.